Okay, thank you very much for the organizers. I'm happy to be a part of this conference, and my name is Songon, but it's simply you can just call me Song. It's easy enough to remember. So today, what I'm going to talk about is something new we've been trying to identify. It's the reversible metabolic complexes in living cell system, which can be possibly being used for the novel drug discovery. So as I always does, I usually start with this schematic diagram of cellular organizations. In order for the cell to be functional and live, all of these compartments, particularly including these organelles, should be communicating with each other in any necessary event. So it's quite clear that this blue the cytoplasm in blue are filled with a number of biomolecules, particularly including the enzyme. So some group of enzymes are responsible for synthesizing building blocks for the cell, like fatty acids, amino acids, nucleotides, etc. And another set of enzymes will be generating the energy and reducing power for the cell. So these processes are called, catalyzed by these groups of enzymes are called metabolism, and whose network is highly integrated by this number of enzymatic reactions, as you can see in this map. So if we go in a little bit detail as part of this map, if we're thinking about glucose metabolism, which is associated with the mitochondria to generate cellular energy, at the same time, exactly the same set of enzymes and the same set of the metabolic intermediate should be shared by the biosynthetic pathway to generate biomass. So the cell should have to know how to balance between energy and biomass production using the same group of enzymes. So in other words, cells should have some sort of mechanism to direct the flux, metabolic flux at every single metabolic junction, whether this intermediate is going to be used for the energy or the biosynthetic pathway. So with respect to the current paradigm of metabolic regulation, the thermodynamics is a keyword to explain the metabolic pathway and their regulation. In simply speaking, the concentration of metabolite is the driving force to regulate the enzymatic activity. However, if you think about the where and when the thermodynamics controlling metabolic enzyme, spatially well-mixed system is how we think about the location of an enzyme. However, if we're going back in 1970s, people have hypothesized there is a mechanism of compartments of the metabolic enzyme. What does that mean? If a cell has utilizing the same set of enzyme in two different spatial temporal mechanisms to regulate this metabolic plasticity. However, due to the lack of experimental evidence to support this compartmentalization hypothesis in literature, current paradigm tells us that spatially well-mixed enzyme is controlled, somehow know when they need to work with which downstream enzyme in which location in subcellularly. So it's been quite open question in this field. So one of the pathways we've been studying is the human purine biosynthesis. Briefly, this 6 enzymes in this metabolic pathway, 6 metabolic enzymes, including 3 multifunctional enzymes, catalyzing 10 chemical reactions to synthesize inosine monophosphate as a precursor for the AMP and GMP. So as you can imagine, based on the discovery of the book, published in 1972, this biosynthetic pathway has been well characterized and documented a long time ago. So pathologically, this purine metabolism has been already known to associate with various human diseases, including leukemia and hyperuricemia. At the same time, at least 9 metabolic syndromes are caused by genetically caused by the inborn error of this purine metabolism. So accordingly, the scientists have been working hard on this pathway to target, and research on anti-metabolite or purine have led to a number of small molecules, including the small list shown in here from the 1988 Nobel Lecture Series from the in Nobel lecture by the Dr. Aliens. But at the same time, even though what does this mean to us that inhibiting or regulating one specific enzymatic activity have been successful in terms of drug development with respect to purine biosynthesis. But biochemists have been more interested in understanding this pathway further, particularly including this trifunctional enzyme. As a point of the biochemist, if you look at this trifunctional enzyme catalyzing non-sequential 3-step 2, 3, and 5, have been paid attention along with the enzyme catalyzing step 4. So today, you don't need to memorize all this name of enzyme. I will just go over simply. But along with respect to the protein-protein interaction and substrate channeling in terms of controlling the metabolic flux, the pair have been quite a bit, got a lot of attention by the biochemists. And late 1970, if you look at these two publications, unfortunately, there are only two publications, as far as I know, have been reported the co-purification of this highlighted metabolic activity belong to this pathway, have been purified from the pigeon and chicken liver. So these two publications in late 70s has sparked the possibility that this enzyme may form a metabolic complex or a metabolic compartment in living system. But as I can tell, since 1980, there is no experimental evidence to support direct protein-protein interaction between these two enzymes and even among all 6 enzymes involved in this metabolic pathway. So since in vitro study had not been very successful, so we move into the living cells to directly monitor the dynamics of this enzyme under the fluorescence microscopy. So we tagged enzyme of interest belong to purine biosynthesis with green fluorescent protein, and we introduce into the living cell, we watch where they are. But as you can see, these enzymes, the GFP tagged enzyme didn't produce any interesting phenotype when from the cell grown in standard culture condition. But if you're thinking about the pathway I'm studying, it's a de novo purine biosynthetic pathway. So if we force the cell to synthesize purine from the scratch, the cell may upregulate this group of enzymes. So we designed the experiment to get rid of the purine precursor or purine from the cell culture condition. Then we start to see our target enzyme start to form this fluorescent cluster in the cytoplasm. After verifying colocalization of all the 6 enzymes involved in this metabolic pathway, we propose the formation of purine synthesizing complex, which is, we call it the purinosomes. After supporting, supporting the hypothesis of purinosome by so many different methods, but most importantly, the purinosome, the fluorescent cluster formed under the purine depleted condition, was dissociated as a function of time upon the addition of purines. Subsequently, when we restored the purine depleted condition, this fluorescent cluster representing the purine synthesizing complex were formed again in the cytoplasm, and this reversibility represents the functional activity of this fluorescent cluster. And this notion was also supported by conventional radioactivity assay using the C14 glycine. So if you use pulse radioactively labeled glycine, this will be part of the purine being become the part of purines synthesized by the de novo purine biosynthesis. So simply speaking, the rate of de novo purine biosynthesis was about 42% increase relative to the cell maintained in purine rich condition, in other words, standard culture condition. In addition, we also characterize that the purinosome in red in this image is surrounded by or harbored by the microtubule network in green. So we believe that microtubule somehow control the spatial distribution of the purinosome in living cell system. So this is one of the reasons why we will not be able to see this metabolic complex or protein-protein interaction in outside the cell. So since then, and we and others have been elaborating this purinosome and then the regulatory system of purinosome using the living cell. And so this summary slide has, with the references, is indicating our current understanding of purinosome. But due to the time I have and also the nature of this conference, I'm not planning to go into the detail about this purinosome in my talk. However, during the conference, if you simply are interested in knowing more about the purinosome or this purine synthesizing complex, I'll be happy to chat with you. And at the same time, one may wonder whether the phenomena like the purinosome is going to be rare in nature or it's common in nature. So now I need to take you back to the 1987, Dr. Paul Soretti, currently in UT Southwestern Medical Center, wrote about the review of the complexation of sequential metabolic enzyme. So the table 1 in this review is pretty much represent the essence of this review. So based on what we know now, except for the DNA, RNA, protein metabolism, and this table 1 was unfortunately over-ambitiously constructed, because if you go into the look at the real cited references, it doesn't give you much of the insight about the metabolic complexes. It may give you the one specific example of protein-protein interaction or the small kinetic enhancement in the presence of other enzyme. However, it didn't cover the entire group of the enzyme for the given pathway. But at the same time, if you are in this field, the references in this review can motivate you to investigate whether this complex really exists in living cell or that this is going to be some speculation. So personally, I have been spending a lot of time on this glucose metabolism. Briefly, the glycolysis breaks down the glucose into the pyruvate by 10 enzymatic reactions, and depending on the oxygen state, it will convert to lactate, or it will move to the mitochondria for the energy, ATP. But interestingly, cancer cell significantly remodeled or reprogrammed this pathway, and well-received example of this reprogrammed molecular player, including hexokinase 2 isoform being outer membrane of mitochondria, and then also phosphofructokinase catalyzing step 3 in this pathway being glycosylated, so that it's inactive, so the metabolic flux is going to be shown to the pentose phosphate pathway. And at the same time, pyruvate kinase M2 isoform being dominant in cancer, and which end up blocking the step 10. So consequently, the serine biosynthesis being upregulated. So pretty much, if you look at this slide, primary goal, if you consider the glycolysis, the primary goal of the glycolysis for the energy production, in cancer cell, it's not. Cancer cell, the primary goal of the glycolysis is to support biosynthetic pathway rather than energy production. Then the alternatively, people have been approaching to understand this energy production through the glutamine biosynthesis, which is not the topic of my scope. So, but what does this mean to me, to us, is that this metabolic plasticity we saw in this glucose metabolism from the human disease like cancer, which made us hypothesize there may be the self-assembled metabolic complex between for the enzyme involved in this glucose metabolism, just similarly to the purinosome. So in order to investigate this hypothesis, we again have been trying to tag our protein of interest with green fluorescent protein and or the red fluorescent protein. And then we're trying to put it into the live cell. We're trying to observe this enzyme of interest directly in living cell systems. So excitingly, the image doesn't come out very well, but the the fluorescent GFP tagged enzyme onto enzyme actually generated this fluorescent cluster look like the purinosome. At the beginning, we confirmed this phenotype in the HeLa cell, but later on, now we know that this phenotype also exists in breast cancer cell and also 2 different pancreatic cancer cell lines we got from the Johns Hopkins University. After that, our colocalization study have been confirmed that all of the cytosolic irreversible enzyme regulating glucose metabolism have been verified to be part of this verified as a part of this fluorescent cluster. So we propose there are at least some group of enzyme in this pathway being a pathway from the metabolic complex, which currently we name as the glucose regulating complex. Most importantly, we have also seen the mobility of the enzyme involved in this cluster. At the same time, we did see the transient interaction between the enzyme participating in these complexes. What this mobility and transient interaction indicate us that this complex has a functional role to regulate the direction of the glucose-derived carbon flux. And obviously we have more experimental evidence to support this possibility of the complex formation, but we also accidentally identify that the clustering formation, clustering phenotype got significantly improved in the presence of resveratrol, which has been quite a debate in the scientific community. At the same time, it looks like whether whatever the mechanism of action of resveratrol, it may still have the benefit for our health. But interestingly enough, this resveratrol induces clustering, and then at the same time, when we get rid of this resveratrol from the condition, unfortunately, the light doesn't go well with this my presentation, but this cluster was dissociated as a function of time. So this slide is briefly introducing you that this fluorescent cluster we have observed indeed reversible with respect to the in the presence and the absence of small molecule, including resveratrol. At the same time, we also have the data utilizing the small molecule activating one of the member of this cluster called pyruvate kinase M2 activator, which also does the similar phenotypic induce the similar phenotypic change. So in summary, personally, I believe that there are at least 2 distinct cellular bodies exist in human cell, including the purinosome and glucose regulating complex. So based on this biochemical map, we know that these 2 pathways are closely related to each other. And the branch of the glycolysis providing 2 molecules, which is the starting material for the purine biosynthesis. So indeed, biochemically, these are really close. So we hypothesize if these 2 pathways biochemically so important each other, these 2 unique cellular bodies we have identified in the living cell may have similar spatial temporal relationship. So we transfected 2 enzymes with 2 different colors representing each metabolic pathway. And then when we co-image in the living cell, we can see that this green and red are not in the same spot. They are different cellular bodies. So we are talking two different cellular compartments, metabolic compartment for the 2 different metabolism. And at the same time, it's quite interesting to notice that these 2 cellular compartments, metabolic compartment, make a pair in space. So seemingly, it is supporting their metabolic relationship in space as well. So I would like to wrap up my talk by giving some sort of insight at this moment. And if we look at it, how people trying to develop the small molecule drug, historically, people trying to directly inhibit, or activate, or allosterically control the specific metabolic enzyme, including the isocitrate dehydrogenase is one of a big deal currently, and also pyruvate kinase M2 related to this metabolic pathway. But this, one of the downsides of the developing small molecule targeting the specific enzymatic activity, it may also affect the same enzyme in normal healthy cell, so that it will generate side effects and also including cytotoxicity in part. So what we propose to the community that there are the metabolic compartment exists, dynamically exists depending on the environment, and if we can generate the small molecule enhancing the complexation or enhancing the dissociation of these complexes, this small molecule may generate a new class of the small molecule regulating metabolic complex. What does this mean? We're not attacking or we're not regulating specific metabolic enzyme. We're just attacking the complex of this enzyme so that the flux will be influenced by the small molecule. However, normal cell, which do not have this complex, is not going to affect it by this small molecule. So without further ado, I would like to thank my group. The postdoc, Dr. Kyung, has been the one of the driving force to move on to my independent career to studying the glycolysis and the purine biosynthesis. And then I have 3 graduate students in my lab. And one of the mission of the University of Maryland, Baltimore County is to incorporating the academic research with the undergrad students. So there I have been had a very valuable undergrad student in my lab quite a while. And then with respect to the small molecule, as I mentioned, at this moment, it may be speculation. It may be, I'm hoping I'm hoping for the luck, but we initiated a collaboration with Dr. Jim Ingley, who directs a high-throughput screening facility in NIH. And this, in the background, if you see this green cluster, this cellular body will be the cellular readout to monitor to screen the small molecule in the future. So thank you again for the invitation, and I'll be happy to answer your question.
This is a nice evidence to support the earlier talk by Dr. Weber, who emphasized the importance of looking at the gene expression profile as a potential target for tumor, site-specific tumor. And what you're saying is that we should look at intracellular complex, not the enzyme molecule themselves as a potential target for drug development. I think there's something common, and I'll have similar conclusion to emphasize on Wednesday morning in my own talk.
Okay, that sounds wonderful. So from the Dr. Weber's talk this morning, so the scientists who are interested in metabolomics or transcriptome or all these different layer of the complexity. So in my case, I'm trying to push this particularly metabolic compartment exists in the protein level. So if this protein level may be explained by the under the different level, including the messenger RNA level or gene expression level, we don't know. However, this complexation, this compartment may be unique in certain specific cancer or specific disease model. So that's what we are currently working on.
I think that it's unfortunate that the first speaker today didn't show up, because you see his topic about water movement across cell membrane could be implicated in complex formation inside the cell, you see, so that the cell may have at least 2 ways of regulating metabolism. The local metabolism through enzymes, say global metabolism by the cell as a whole, and one of them is water movement. If you hydrate or dehydrate, you can form or inhibit complex formation.
I think that's a really good point. And so the reason I'm trying to develop this metabolic characterized metabolic compartment for the glycolysis, this the relationship between the purine biosynthesis and glycolysis as a small minimal network, so that we can understand this how the cell can utilize this all complicated network of metabolism in the future. So there might be a lot of possibility. So I wanted to present there is such a thing out there to this new community So that's what I mostly appreciate, having a chance to give a talk in this conference. Thank you very much.
Yes, give applause to
Thank you. Oh, thank you very much.